Neurotransmitters, along with electrical signals, are the means by which each neuron communicates with others to process and send messages to the rest of the body. Neurotransmitters can be classified chemically and functionally. Let's first classify the neurotransmitters by their chemical structure. Neurotransmitters fall into several chemical classes based on molecular structure. There are conventional and unconventional neurotransmitters. Conventional neurotransmitters are stored in synaptic vesicles. They are released when calcium enters the axon terminal in response to an action potential. They act by binding to receptors on the membrane of the postsynaptic cell. They include small molecule neurotransmitters such as amino acid neurotransmitters, glutamate, GABA, and glycine, biogenic amines, dopamine, norepinephrine, epinephrine, serotonin, and histamine. Purinergic neurotransmitters include ATP and adenosine and acetylcholine. Some neuropeptides, such as endorphins, enkephalins, substance P, and neuropeptide Y, are also conventional neurotransmitters. They are made up of 3 or more amino acids and are larger than the small molecule neurotransmitters. Unconventional neurotransmitters include endocannabinoids and gasotransmitters. Now let's classify the neurotransmitters according to function. Neurotransmitters can be classified by their effects, inhibitory versus excitatory. They can also be classified by their action, direct versus indirect. Now let's look at the receptors. There are 2 types of receptors, ionotropic and metabotropic. Ionotropic receptors are membrane-spanning ion channel proteins that open directly in response to ligand binding. Ionotropic receptors are large protein complexes with specific binding sites for neurotransmitters. When the neurotransmitter binds to the receptors, the protein complex changes shape, causing the channel to open. Ionotropic receptors may have either an excitatory or an inhibitory effect, depending on the ions that can pass through the channel and their concentrations inside and outside the cell. They typically produce very quick physiological responses. The current starts to flow within tens of microseconds of neurotransmitter binding, and the current stops as soon as the neurotransmitter is no longer bound to its receptors. In most cases, the neurotransmitter is removed from the synapse very rapidly. An example of an ionotropic receptor is the nicotinic cholinergic receptor. Metabotropic receptors trigger signaling pathways that may indirectly open or close the channel, or have some other effect entirely. The activation of the second class of neurotransmitter receptors only affects ion channel opening and closing indirectly. In this case, the neurotransmitter receptor is not an ion channel. Signaling through these metabotropic receptors depends on the activation of several molecules inside the cell, and often involves a second messenger pathway. Because it involves more steps, signaling through metabotropic receptors is much slower than signaling through ligand-activated ion channels. Some metabotropic receptors have excitatory effects when they are activated, while others have inhibitory effects. Often these effects occur because the metabotropic receptor triggers a signaling pathway that opens or closes an ion channel. Alternatively, a neurotransmitter that binds to a metabotropic receptor may change how the cell responds to a second neurotransmitter that acts through a ligand-activated channel. Signaling through metabotropic receptors can also have effects on the postsynaptic cell that don't involve ion channels at all. An example of a metabotropic receptor is the muscarinic cholinergic receptor. If you like this video, please hit the like button below and subscribe to my channel to see more videos in future. Thank you for watching. Bye.